I am Chris Holzapfel. I'm the Holzapfel part of the Bradshaw-Holzapfel Laboratory of Evolutionary Genetics. I've spent the bulk of my research career at the University of Oregon, having come here directly from my postdoctoral work at Harvard University, bypassing a tenure-track position at the University of Michigan, which I will admit was not the easiest decision I've ever made, but in hindsight it was a really good one. Bill Bradshaw and I have been able to develop a novel system together in which we have been able to answer transformative questions for more than 30 years. Today we are going to talk about the work that is ongoing in our research right now. We have 3 arms of research that we are focusing on. One is on the eradication of blood-borne disease. The other is on biological timing mechanisms, that is the integration between the daily timer that actually allows you to get up in the morning and the seasonal timer that tells us about how the world looks around us. And finally, climate change and the observations that we made relating to climate change penetrating to the level of the gene and how that changed the understanding of that phenomenon. So what is this novel system with which we are working? It is the pitcher plant Wyomia smithii system. Wyomia smithii is a little black mosquito, kind of nondescript, small, that lives only in the water-filled leaves of the purple pitcher plant, Sarracenia purpurea. This is a carnivorous plant. I'm actually surrounded by them here. And this mosquito spends its entire pre-adult development and life in the leaves that normally eat insects for breakfast, lunch, and dinner. So for 2 young scientists starting out, it seemed like this had to be a system that was going to tell us many interesting things. Of course, when you start working in a system, you don't know much about it. We've learned lots of things. I'll just tell you a couple of them today. First of all, it extends over a huge geological and geographical range. It occurs from North Florida in the south all the way up into northern Canada, so it has a big latitudinal range, and it also occurs from sea level to the Appalachian Highlands in North Carolina, so it has a good altitudinal range as well. We have learned about the phylogenetic relationship between different populations, and I said we have over 100, at the level of the nucleotide. It's difficult to do genetics, particularly if you're doing genetics in nature, unless you know the relationship at a genetic level between your populations. As you might have gathered, Bill and I are interested in how genetics actually works in nature. We're not interested in how it might work in nature. We want to use Wyomia smithii to find out how it actually does work in nature. Anyone that knows me knows that I'm applied to a fault, and that's just the truth of the matter. So I am going to talk about the eradication of blood-borne disease. Every minute a child dies in just sub-Saharan Africa from malaria. And this is not counting the many other diseases that are also carried by mosquitoes. Dengue, yellow fever, many encephalitises that cause human brain diseases. Right now, there are thousands of labs worldwide that are doing really heroic work, determining ways to mitigate blood-borne disease, trying to figure out vaccines that might work. All of these labs are doing one thing in common, and that is they're all assuming that a mosquito bite will occur. So we took our system of Wyomia smithii that lives in this pitcher plant and said, okay, so what can this system tell us that's different? Bill and I have known for 20 years easily that Wyomia smithii is the only mosquito that has been described out of the thousands of mosquitoes that are known that bites in one part of its range and is an obligate non-biter in the rest of its range. So we are able to actually extend the discussion from the level of biting to what happens if there is no bite. If there is no bite, there is no blood-borne disease of any kind that is transmitted, period. So no bite equals no disease transmission, period. The secret that we're looking for is held somewhere in the middle. The genes that are involved in the biting phenotype are somewhere on the transition from biting in the south to obligate non-biting in the north. What are these genes? So the first thing that we did before we started on this project was to talk to our colleagues about it. And they said, well, clearly what you guys are doing is you're working with two different species of mosquitoes here, a non-biting mosquito and a biting mosquito. So we went into the lab and we did reciprocal crosses between the northern non-biters and the southern biters, and we found, simply put, that the northern males actually way preferred to mate with the southern females and vice versa. So we're working with one species. They produced fertile offspring. That's the definition of a species. Then there were other colleagues that said, okay, well, during the evolutionary expansion of Wyomia smithii, what has happened is that the northern animals have lost their biting mouth parts. Possible. We had a graduate student a couple of years ago that looked at electron microscopy, scanning electron microscopy at a very detailed level, and found that the biting mouth parts of the northern Wyomia were perfectly capable of taking a blood meal. They simply don't do it. So we have multiple arms of research going on in our lab. So needless to say, we're going to use multiple ways of addressing this problem. We have students that are working with quantitative genetics. During the 3 or so years that these students have been doing these experiments, they take forever, we have been also developing what is called a voracious biting line. So what is a voracious biting line? It is a line in which the genes that are involved in the biting phenotype are concentrated. So what will we do with this voracious bloodline? We have one group of colleagues that are working on what is called comparative genome studies. A genome represents all of the genes that are present in a population that you have sampled. One genome from the non-biting north compared with a genome from the voracious blood-feeding line from the south, with an eye towards allelic variation between those two. We have another group of scientists, including ourselves, that are working with differential gene expression. So what is differential gene expression? It tells us about details of which genes are turned off, on, up, or down regulated as relates to your specific phenotype. In our case, our phenotype is biting. Once we get the number of genes to about 100, then we are going to work on the verification of those genes using a technique called RNAi or RNA interference. And basically in gene verification, the genes that are intimately involved in the biting phenotype are kept, and those that are supplementary to the biting phenotype or less deeply involved in the mechanism are not kept. We already have several pharmaceutical companies that are interested in developing non-toxic inhibitors that will change effectively biting mosquitoes into non-biting mosquitoes, and that will be our next step. And if those pharmaceutical companies are able to succeed in this process, we will have changed the direction of medicine for thousands of people worldwide. Are we definitely going to succeed in this process? Absolutely. Mother Nature did it once. All we have to do as a group of scientists is to figure out how. Before I turn this over to Bill, I do want to say one quick thing, and that is that if you're actually working right on the cutting edge of science and are asking a really good question, a lot of other labs are going to be asking that same question. Thus, what we always do at meetings and the like, we find out how are other labs approaching this question, and then we do something different. We take our mosquito system and say, okay, Wyomia smithii, what can you do to tell me what is different than what everybody else is doing? if you aren't asking something in a different way, it's very unlikely that you are coming up with a new solution to a problem that will truly transform an area and move discovery forward.